the torsion angle or the dihedral angle. It is something complex. So the torsion angle or the dihedral angle, it originates from the three-dimensional space angle idea. That means, let's imagine H2. That means here one hydrogen, it joins to another hydrogen by a bond. So it can be shown as this type. That means here the stoichiometry of H2 is defined by the distance, that means bond length. But let's come to the water. In that case, the stoichiometry defines by two factors. That means one is bond length or the distance, another is the angle. Or this angle is important because of the two factor, that means distance along with the angle. This molecule is present in the three-dimensional space. It cannot be present in the two-dimensional. But in the H2 case, because of the presence of the one factor, it can be present in the two-dimensional. But when any angle is joined along with the distance, then it can form only in the three-dimensional case. For example, in the H2O2 or the hydrogen peroxide, here it is the one angle, here another angle, here it is the distance of the bond length, here bond length, here bond length. That means many factors are involved. That's why this is called, that's why they are present in the three-dimensional spaces. For example, this angle, this is called the torsion, and this angle is called the torsion, this angle is called the dihedral angle. So these angles are called as the torsion angle and the dihedral angle. So always, because of this three-dimensional space, these angles are formed in the planes instead of the lines. In this case, the angle can be present in the planes. That means simply we can say that 180 degree. But here, this angle should be present in the plane. It cannot be formed in the lines. So like that, this is the protein of the peptide. Here it is the bond between the N and the C alpha. Here N and the C alpha. It has the angle that is called the phi. It is designated as this. So there are different types of the angle. This is called the phi. And the angle between C alpha and C, it is called the psi. And psi is designated as like this. And angle between C and the nitrogen, it is called the omega. So there are different types of the angle, and from this angle we can determine that which molecule can be formed the alpha helix, which molecule can form the beta helix. For example, this is called the Ramachandran plot. That means in the Ramachandran plot, at the x-axis the phi is present and the y-axis the psi is present. The phi ranges from minus 180 to plus 180. What is plus 180? That means when the degree of rotation is clockwise, then it is called the positive. When the degree of rotation is counterclockwise or anti-clockwise, then it is called the negative. So like that here also. So here ranges the minus 180 to 180. Here also psi ranges from minus 180 to plus 180. Let's see this dot. This is the anti-parallel beta sheet. We will know about after some time. So that means the anti-parallel beta sheet has high psi degree and low and high minus phi degree. For example, this anti-parallel beta sheet has exactly minus 139 phi and plus 135 psi. So different molecules have different angle of the rotation, and by knowing this angle of the rotation we can determine whether it is alpha helix, whether it is anti-parallel beta sheet, whether it is parallel beta sheet, like this. So again one thing, that this bond, this has a partial double bond character. So because of this partial double bond character, it cannot rotate the angles properly. That means it hinds the rotation. So that's why this phenomena is called as the steric hindrance. Again, let's say in this molecule, this is one amino acid, this is another amino acid. Here this R group is hydrogen, that means it is most simplest. For example, if in the peptide this is the proline, if the proline is included, then it has, this is the structure of the proline, that means it has R group is like this. This is the R group and this is more complex. So because of this complex or the bulky R group, it cannot allow the rotate of the angles. So it will hind the rotation. So again it will form the steric hindrance. So that's why in any proline rich people, in proline rich protein, the degree of the rotation will less. But if the glycine is present in the protein, then the freedom of rotation is very high. For example, this antiparallel beta sheet, it restricts only this area. This is the parallel beta sheet. It restricts only this area of the degree of the rotation. But the exception is the glycine. The glycine has the most simplest R group. That's why it has a very high range of the freedom of the rotation. That means it can present here, it can present here, it can present here, it can present here. So it can has a very high range of freedom of rotation. So this is in the Ramachandran plot. This is the antiparallel beta sheet. This the range is -139 and psi is +135. Like in the parallel beta sheet, -119 and +113. So always this torsion angle or the dihedral angle, it determines the interaction of the molecule in the protein. That means this angle determines whether the protein is going to be an alpha helix, whether it is going to antiparallel beta sheet or the parallel beta sheet, and like that. And it is determined by this Ramachandran plot, and it was discovered by Ramachandran et al. Obviously, as name suggests. One more concern I want to share with guys. Many guys are watching my videos, but most of are just watching only, but not subscribing it. So please subscribe my channel so that I can boost myself and I can bring much more videos like this. Okay, bye.